Welcome, everyone. Can you hear me? Okay. So it's my pleasure to introduce to you your speaker for this morning, Erik Segerdell, who is a master's student in DMICE. And Erik received his bachelor's degree in biology from the University of Oregon in 1992. And then he moved on to working with the zebrafish model organism database. He was a scientific curator and ontology developer for them. And currently, in addition to working on his master's degree, he works on two separate projects. He's a developer for the Quantitative Imaging Pipeline Informatics Working Group in the Knight Cancer Institute, and he's also involved in what's called the ZenBase project, which is based in Cincinnati and is an online database of frog genetics and genomics. And this morning, Erik is going to give his pre-proposal defense. He's going to talk to us about how can an interactive visual analytics tool help biomedical scientists investigate genotype-phenotype relationships.
Okay, good morning. Everyone can hear me okay? And thank you, Beth. So yes, I will present my thesis project proposal. It'll be a study of how an interactive visual analytics tool can help users investigate relationships between genotypes and phenotypes. The objective of this presentation is to first give you some brief examples of how we view biomedical data in informatics tools and databases, specifically data related to genotypes and phenotypes, and illustrate a challenge that we face. I will then give an overview of the specific aims of the project, followed by my proposed plan for achieving those aims, which will consist of prototyping and evaluating user interface features in a visual analytics tool. I'll talk briefly about the expected outcome and the hoped for benefits of the study, followed by a proposed timeline for doing the work. Now, seeking biologic... Yeah. No, it's supposed to wrap. Okay, that's better. Thanks. Technology. Okay, seeking biological insights into the relationship and distinction between phenotype, or the observable traits of organisms above the molecular level, and genotype, or the genetic makeup of organisms that is inherited from their parents, has long been fundamental to many areas of life sciences. Today there is a vast and growing wealth of information about genetics, genomics, phenotypes, embryonic development in a variety of biomedical and model organism databases. And these data present researchers with a great opportunity to uncover human disease mechanisms and to drive discovery of new therapies, provided that they are able to make sense of extremely large amounts of information. Currently, we lack suitable bioinformatics tools to enable investigators to effectively meet that challenge. There are visual analytics approaches, however, that can provide insight and inspiration that will make these interconnected data better understood. And what I want to do is investigate how interactive user interface features can help users investigate genotype and phenotype data and begin to synthesize insights from what they find. And my goal is to identify and test a set of interactive features, or user interface elements, that have the potential to enable investigators to navigate this kind of data in a web-based interface in such a way that they can start developing mental models and hypotheses about underlying human disease mechanisms. What I propose to do with guidance from two initiatives involving bioinformatics research groups at OHSU, whom I'll talk more about in a bit, is to carry out a qualitative assessment and usability study of an interactive visualization prototype, the design of which will be based both on these groups' expectations and needs for the visualization tool, as well as through user interface requirements analysis. Now, there are a lot of resources and interfaces out there already for this kind of data. I'll present a couple of examples. Model organism databases have been around for a relatively long time, and I've worked as a curator for a couple of them. And they're wonderful resources that contain an enormous amount of genetic, genomic, phenotypic data about animal model species like the zebrafish and mouse and rat and frog. Their web interfaces are primarily text-based, as you can see in this example, where you may have to sift and click through a lot of pages to find information you want. So it's a challenge to navigate all of these data and all of these types of data if you want to form some overall picture of the biology associated with whatever it is you're interested in, be it, say, a gene or a phenotype. This is something that's been developed more recently. It focuses on one type of data. It's a visualization widget developed by the Monarch Initiative, which is a project I'll discuss a bit more shortly. It's a phenotype comparison grid with the phenotypes associated with a human disease listed in the rows. The human diseases listed in the rows and corresponding mouse genetic models in the columns. The little cells are shaded and colored in order to indicate similarity between the phenotype and the corresponding model. And I don't want to concern us with the details of how this works. It's just intended to provide an example of how you can start to include elements like color and filtering and interactive elements to explore these data in one place. And what my project entails fundamentally is exploring how these kinds of dynamic interactive elements can be applied to making sense of the great variety and volume of related biomedical data that I touched on the previous slide.
So is this looking at like phenotype that's comparable between mouse and humans, or is it looking at, we know the gene that causes this phenotype in humans, and what phenotype does that same gene cause in mice? So is it more you're matching phenotypes or you're matching genotypes on that?
It's matching phenotypes between.
So mice can have like high foreheads, or?
Yeah.
Okay.
Or something
Some
similar.
analogous. Mouse.
Exactly. Yeah.
Okay. Interesting.
So here I want to look at a real example of the interconnectivity of several types of biological data, and it's just to give you a little sense of the richness of the data that's out there. So this is a diagram in the form of a network graph that integrates some human and mouse data about a disease, which is ALS, and related genotypes and phenotypes. Human data are indicated by the light blue circles or nodes. The mouse data are shown as the yellow nodes, and the green nodes represent both human and mouse, so mammalian data. The labels on the arrows tell you something about how the nodes are related. A disease has related phenotypes. Human phenotypes have similarity to mouse phenotypes, and so forth. So you have here a rich set of biological relationships that you can traverse via multiple paths between the human and mouse phenotypes, which are near the top, and the human genotype is down at the bottom. You can see how similar nodes can be grouped, such as phenotypes bound by the boxes. The take-home message here is that this is a pretty informative way of picturing many types of interconnected data related to genotypes and phenotypes, but keeping in mind that even all of this amounts to only partial information about one disease, and it's just a sliver of all the data that's out there. So it's a challenge to make sense of all of these data and relationships and to create this kind of visual picture with existing tools and resources like the traditional animal model databases. So why is that important? It's important because our tools should help investigators form hypotheses about genotype-phenotype associations and how they underlie human disease. Here's another diagram in the same vein as the previous one, except it's more of an abstract example. So imagine that you have a potential path from a phenotype of interest at the top to a disease at the bottom, and that path is incomplete. And it's a path that would be complete if there was a known confirmed direct relationship, or some interaction between the two light blue human genes, number 1 and 2. It may be that the two human genes are known to be in the same genetic or protein interaction pathway, or it may be that the mouse orthologs of the human gene exhibit similar human phenotypes or gene expression patterns. If you have the ability to picture the data in this way and in these contexts, perhaps in one place, then this picture can potentially lead you to formulate the hypothesis that some critical functional relationship between the two human genes exists. So in other words, it's leading you to really start synthesizing the biology and developing a scientific narrative. And I'm going to come back to this when I discuss my project plan in a few minutes. Now, in preparation for my project, I was pointed to some very interesting research by Barbara Morell. This paper is from 2009, but it's still very relevant. She explored how future bioinformatics tools might give scientists the support they need to move beyond conducting queries and vetting results and classifying relationships, and moving beyond that into the realm of forming mental models of the underlying biology where they are formulating hypotheses and telling a scientific story. Her study concentrated on protein interaction networks and tools to view those networks. She found that while existing tools did a good job of supporting tasks related to finding relationships of interest, future tools could potentially do more to support tasks that let users gain what she called causal insights into the biological systems being studied. She referred to these as explanatory analysis. So it's a step beyond exploratory analysis. She identified features in visualization tools that scientists would have liked to have had or liked to have seen more robustly supported, and that might have enabled them to start doing that kind of explanatory analysis or synthesis. And what you see here is not the complete list of interface features that she identifies in her paper. Rather, it's a few examples that provide some inspiration for the sorts of features that I might put to the test in my own project. I'm interested especially in prototyping interactive elements, but certainly some of the content-related features here could inform my designs as well. And finally, before I proceed to the specific aims of the project, there are a couple of local collaborations that are crucial to it. The Monarch Initiative is a collaboration between groups at OHSU, University of Pittsburgh, and several other institutions, which plans to deploy a web application that features semantic tools for users to navigate, manipulate, and interpret the rich content provided by animal model databases that I talked about. Plans to develop a visual analytics software environment for exploring these data. In addition, Beth here at OHSU is interested in, among other things, visualization of functional genomics data. The snapshot you see here on the right is from HitWalker, which is a tool that aims to integrate genomic information with complementary functional analyses in order to prioritize sequence variants in cancer. And so I'm going to lean on these two groups to help me develop specific use cases for visualization and analysis of genotypes and phenotypes, and to help narrow down the set of interactive elements that I want to prototype as part of my work. And I'll also be discussing with them their own expectations and perceptions of what users want from these sorts of tools, because I think it'll be interesting to find out how those perceptions may or may not differ from what users find that they want as they start trying these things out. So that's the overview of the background for this project. I'll move on to the research question and the aims. Now, I've defined some aims to address my research question, which is, how can an interactive visual analytics tool help biomedical scientists investigate relationships between genotypes and phenotype? I talked a moment ago about examples of potential visualization features that tend to be not supported robustly in existing tools, but that may be useful for helping users move beyond exploratory analysis and into explanatory analysis. What I propose is a usability study where I identify some use cases and then specifically select a small set of interface features and then try them out. In the scope of this project, I can't possibly test out all of the possibilities, but I intend to focus on a few features which, working together in complementary fashion, might enable users to start synthesizing biological narratives and form hypotheses. So let's go back to the diagram I discussed earlier. When I speak of investigating links between genotype and phenotype, you might have a tool that represents data like a network graph, if that's beneficial. It doesn't necessarily have to be that. But you might not want to have a display that shows all of the data that you have at once, which could be indecipherable if you have a huge amount of it. Instead, the user may wish to start at one point, like a phenotype of interest, or perhaps a small subnetwork, and then start exploring by following interface cues or being given information by what is connected to it, until they can start to build up this sort of picture of the data. And it may in practice be hard to get all the way to this stage, but the use cases that I'll develop to help answer my research question will likely be along those lines. It may not be this exactly, but the idea is that you might start at one point and then proceed from there and see where you can go. All right. Aim number one is to determine the design of a visualization environment or tool containing the interactive features that I want to test, and to refine the study design. And this will involve user scenario development and initial mock-ups of an interface, along with fleshing out the study design. Aim number two will comprise development of a functional interactive prototype or prototypes based on the mock-ups. Aim number three will be to carry out a qualitative usability evaluation of the prototypes, and to ensure that they satisfy accepted usability guidelines and that users find it satisfying and desirable to come back and use. Okay, so let's look at the project plan for carrying out the aims. Again, aim number one is to determine the design of a visual analytics prototype that will be subject to usability evaluation. And the key questions here are, what are the objectives? In particular, what do the Monarch and functional genomics visualization projects want to provide to users exploring genotype and phenotype data? And what requirements do their projects aim to satisfy? Who are the stakeholders and the potential types of users or user groups? What are the use cases? And which set of interface features do I want to test? And are they technically feasible? And I also want to determine the evaluation criteria, meaning the questions that I want to address and ask users in order to gain insight into what they're thinking as they're using the prototypes. Low-fidelity prototypes will be developed in order to provide the basis for subsequent interactive or software-based prototypes. And what I mean here by low-fidelity prototype is one that is done by hand, either on paper or with a drawing package on a computer, and that gives an indication of the user interface look and feel, but without being fully functional. The approach I want to take with the early prototypes or mock-ups is to develop user scenarios. And this means that entire interface interaction scenarios will be illustrated with one screen for each of the steps that a user will go through as they navigate data, much like you might create storyboards depicting a scene in a movie. So this will be an iterative process. And these storyboards will come out of brainstorming sessions with users as they are recruited for the usability study. And I'll describe those users more in a bit. This is an important step because I think it will help convey to the test users some of the possibilities for what an interface for investigating phenotypes and genotypes can do, and it will help me discover what they want it to do. In particular, I'm interested in the more advanced goals that they might want to achieve with it. And that, in turn, will help me focus on the handful of features that I want to put in the subsequent prototypes. Now, based on the mock-ups and their initial evaluation results, I'll develop a functional and interactive prototype for further evaluation. So that satisfies the second aim. And it will help confirm the technical feasibility of putting the interactive features into practice. It'll be a high-fidelity prototype, which in contrast to the low-fidelity one, will be based on software. It'll be functional. It'll be something that users can actually interact with. This prototype will be constructed using a JavaScript library called D3, or data-driven documents. And a mock data store with a subset of data needed to support the prototype will be obtained, most likely from the Monarch project. And I should add, too, that there may be more than one prototype at this stage, if that's feasible, and if it's warranted by a need to demonstrate a range of possibilities for what users can do. And in the process, I'll aim to follow good general interface design principles. So the desired result here is to put the interactive feature set in action and to use some real genotype and phenotype data from a real project. And with the third aim, we come to a usability evaluation of the prototypes. I propose that this evaluation, which will be based on widely accepted and recommended methods, can answer the questions of whether the prototypes are generally usable and inviting to the user, whether the selected interface feature set is effective in practice in facilitating explanatory data analysis, and whether there are unexpected problems with the interface. And hopefully it'll go a little better than this. The user interface evaluation can be constrained by factors such as timescales and the ease of finding participants. And given the time frame for a master's project, discount usability engineering is an attractive alternative to the more comprehensive evaluation approaches, such as quantitative analyses, that you might apply to an entire interface. So discount usability evaluation depends on a small number of users and a small number of interface features. And for this sort of agile qualitative approach, there are a few established inspection methods that are especially useful. And I'll define and discuss these in more detail shortly. The first is a heuristic evaluation. Second involves a think-aloud method involving a small group of users. And the third is a semi-structured interview that includes a simple usability survey and a debriefing that takes place after the think-aloud sessions. The low and high fidelity iterations of the prototypes will be subject to these evaluations. So I have two users performing heuristic inspection, and a different group of perhaps 5 or 6 users will participate in the think-aloud sessions and in the survey and interview. Let's look at each of these evaluation methods individually. The heuristic inspection will be carried out based on Nielsen's heuristics, and I'll aim to recruit one usability expert and one biomedical domain expert to do the inspection. And these experts will focus on general principles for interaction design that you see here. So Jacob Nielsen, back in 1995, established these general principles for interaction design. And it consists of broad rules of thumb, not specific guidelines. And I won't go into detail about all of these. And they're easy to find online if you search for Nielsen's heuristics. But I do want to say that these guidelines were designed to apply to all kinds of different user interfaces that are out there in the world. So some of these items may be a bit more relevant than others to this project. For example, I'd say that the match between system and real world where you want to make information appear in a natural and logical order, is obviously important for a system that strives to allow scientists to chart a course from obtaining query results to developing a bigger picture of the biology. And in addition to the 10 established points, I will also want to add to this evaluation a few points that are specific to the biomedical domain, since this will be my chance to get feedback from a biomedical expert user. And developing those questions will be part of the work that I'll do. So I don't know what they'll be yet, but an example might be how well does the interface convey the reliability or provenance of the information that's shown, which is a critical point in a scientific context. I'll move on to the next method. The think-aloud protocol will focus on a few selected features and tasks. So this method will help gain insight into what users are thinking as they are negotiating the interface. This evaluation will be conducted with users representing several defined groups, which I anticipate will be students, researchers, and clinicians. And I'll aim to have 1 or 2 users from each group. So in the think-aloud walkthroughs of the prototypes, users will attempt to carry out tasks corresponding to the use cases that will be defined during the prototyping stages. And the goal here is to encourage users to do 2 things. One is to describe aloud the actions that they are taking, and the other is to go a bit beyond that and explain or interpret their actions. This method has been described as one that gives you a window on the soul of your users. So I think it's especially conducive to observing how well they are synthesizing information as they explore it. I also expect that this will gauge how well users are able to efficiently and easily navigate to data of interest and to perform their analyses with a minimal amount of help, and whether the interface effectively presents and communicates biological information. The final inspection method involves a short written survey and a debriefing interview with each user. So this will help capture general measures of the prototype's usability. It's called the System Usability Scale. It's a 10-item Likert scale that provides a global view of subjective assessments of usability, a few examples of which are shown here. The questionnaire will be given to each user in the student, researcher, and clinician groups after they've had an opportunity to use the prototypes in the Think Aloud sessions. And after this, I will do a debriefing interview as well to explore each user's reasons behind their answers. And there is a formula to convert the responses to a scale between 1 and 100. And based on some research and further experimentation with this approach, it's been proposed that a score of at least 68 to 70 or thereabouts corresponds to the interface being user-friendly. And as with the heuristic evaluation, I also want to add a few biomedical domain-specific questions. And again, the specifics of these questions will be determined as part of the work. But the intent is to help evaluate the prototype's usability as specifically scientific tools. So what I've proposed is that visual analytics tools can be leveraged by users to perform explanatory analysis of genotype and phenotype-related data, and to move beyond exploratory analysis and begin to synthesize a scientific story. So the outcome of this study will be a report of my qualitative analysis of the prototypes and their interactive features. I also want to produce a case study and provide an in-depth examination of how one of the users in the study actually used the tool and what worked for that user and what didn't work. I think that'll shed light on what users need and how those needs compare to our assumptions going into this. And I hope to come away with some recommendations relevant to future interface development and quantitative evaluations of a complete system later on. And I hope that this study will have a positive impact by contributing to future progress of tools where researchers and clinicians will be able to decrease the difficulty associated with complex biomedical problem evaluation and to fully realize the potential benefits of the wealth of data that's already out there and already exists. All right, so that's my project plan and expected outcome. I'll briefly go over the proposed timeline for the work. The table is broken out into the specific aims. Aim number 1 involves, again, the initial mock-ups and scenario development, recruitment of users, fine-tuning of the study design and the evaluation protocols, as well as IRB approval for usability testing. It would take place over the next month and a half. I anticipate that Aim 2, which is the development of the functional prototypes, to require a month of work. And Aim number 3, the usability evaluation, would proceed to approximately the end of June. And the goal ultimately is to defend and finish by the end of the summer. And finally, a big thank you to my thesis advisory committee, Beth, my thesis mentor and colleague in the Night Cancer Institute, Melissa and Michelle here in DMICE, and Harry at the University of Pittsburgh. And my thanks also to colleagues at the Monarch Initiative, at which Melissa and Harry are co-PIs, as well as those at the BISR and ODG. And thank you all for watching and listening. Good. Yes.
Thank you. An excellent presentation. It's a very complex topic. The question I have is what on your user interfaces, whether or not the emphasis is actually on the usability of the system or the presentation of the final, complete, very complicated data in a meaningful way.
It's really a blend of both. We obviously want more effective tools, but we also want to make sure they go hand in hand with the accepted guidelines that are out there. And I think what's especially important is for these kinds of interfaces to be appealing so that people will want to come back to them and use them again later on.
Who are your target users for this? I mean,
Mm-hmm.
Basically use a tool that was kind of at a different level and asking different questions. So do you have a specific user that you're trying to work with to develop your prototype?
Well, again, it could be a variety, but it's really, I mean, any biomedical investigator looking at the underlying basis for human disease, and researchers or clinicians who are going to model organism databases to try to find models for what's happening in human, and then trying to make sense of all of that. Yes.
I've noticed you selected D3 as your primary tool for building an interface, but I was wondering what happens if you cannot fit the affordances of the D3 development tool to the design constraints that you might discover in Aim 1?
That's something I want to consider actually during Aim 1, where I'm selecting the features. I'm going to try to be mindful that it's something that's technically feasible, because at the same time I'm really going to be having to learn a lot about D3 on the fly. So, yeah.
Just a follow-up.
If I can just follow up on that, that's actually a really complex question. And we've run into a number of different issues with that under the underlying database, how to format the data, there's actually, that's a huge thing. But we have been working on it, and the Monarch Initiative have been working on it, so Erik's not coming in this cold without any help. But it is a really tough problem.
I guess just as a follow-up, this is more of just checking on things, but what will you do if you're not able to design an appropriate interface for AIM-3? Because you have some dependencies that may pose problems if you have barriers along the way.
Right. At the very least, we'll have the low-fidelity mock-ups, which are just hand-drawn, or I'll use a drawing tool actually for that. They'll be subjected to the evaluation as well, and hopefully I'll be able to transfer that to the more interactive functional prototype. But even if I run into a lot of unanticipated trouble with the more interactive software prototypes, we'll still have the more crude prototypes that can be subject to all of those different kinds of evaluations. So you can still get some insight into what users will want to do and what they need to do, as long as the technology can support it.
And that's a really good question, too. We run into issues all the time about that, depending on what data type you're looking at. There's a wide variety of different data types, and it's the value, really, of working with both the Monarch and functional genomics data, because they're different and they're coming at it from a different perspective. But again, Erik's going to be working with a subset of the data, and we're interested in Erik's project will be features, individual features that we want to test. So it's not so much going to be a problem in actually putting together a high fidelity prototype, because we already have that working. What we have found is really some of the queries that we want to ask with some of the data types turns out to be take too long and be more computational problem.
Yes.
I noticed you mentioned you'll be doing a case study as part of your output. And I was wondering if you'll be bringing, your users will need to be, solving a problem or answering a question. I was wondering if you were going to specify that for them, or if you're going to let them organically bring their own to the evaluation.
It'll come out of discussions with users as we recruit them, but then also discussions with Beth's group and the Monarch group. So it'll come organically out of all of that initial work.
So you'll be working with both the Monarch Initiative and group, right?
Mm-hmm.
So the users are going to both.
How do you envision this work being used by both groups?
Do you think both groups will
Want to implement the prototype, or do you think it'll be more targeted towards Monarch or Bitblocker? Do you have any idea?
I think I want to achieve something that it's kind of has a crossover between what the two groups are looking for and need, but not something that's so generic that it's just going to be kind of irrelevant. So, I hope that answered the question. Okay.
Thank you.
Thanks.